what I want. I want a single page. Yes. We have Iram and Rabia also joining in. Good evening, Iram and Rabia. How are you?
Fine, sir. How are you?
I'm very well. Can you see the screen that I'm sharing? This is your NCRT only.
Yes, sir.
Better be continuous. Okay. So I have to show you certain things. So that's why I thought of sharing the NCRT since this is not copyrighted and go through this. Okay. Okay, cool. Okay, so some things that are asked in boards and also in entrances are very, very, trivial and detail oriented from your books only. So I just wanted to, I'm not sure how many of you read the whole NCRT chapters. How many of you do read the full chapter on your own, or do you just rely on, the notes in the classes in your school and my notes? Those who read the whole chapter, like line by line, just raise your hands. Aisha does that. Rabi also does that. Iram does that too. Okay, so Mariam is like, yeah, Parker is like me. When I was in school, I was not in a habit of reading the chapter, the whole chapter, which I realized later on. So, you can also learn from what mistakes other people did. So it's not a mistake in terms of you understanding science. You can understand science even by a YouTube tutorial, or you even by talking to a person who's in science, visit someone's lab and etc. Aisha, you can put down the hand. I understood. Cool. Thanks. So I was also not in a habit of reading the chapter as a student, but then I realized the power of it when I was preparing for entrances. And NCRT is a very good textbook, not just for your undergrad entrances, but also for entrances and at master's level. Okay. So I just thought of, because this chapter and the next chapter, which is molecular basis of inheritance, are the two very crucial chapters and very heavy, bulky chapters in terms of marks and portion that they acquire in the syllabus. So I thought of taking you through the NCRT, and we will learn how to efficiently read your NCRTs. Okay. So those who read the NCRTs, just let me know if you already do these things, which will be great. And I think I can provide a lot more to Parker. Okay, Parker. So whatever you learn in today's class, you have to implement it in every chapter that we are going to study. Is that okay, Parker? Since you were very honest in telling me, oh, wait, maybe she told me in the chat. She wrote, yes, sir. Okay, Parker, do you also read the chapter line by line? Uh, did I? No, okay, fine. So you like me. Great. Okay. No, I'm not happy because you don't read. I'm happy that I got you correct. You should read, and I'm going to tell you how to do that because I also learned it the hard way and it helps. It's very, very beneficial. Okay. So in this chapter, we'll first cover a few important things. Then I'll come back and tell you what things you should pay attention to in every chapter. So this portion we have already covered, right? Inheritance of, so NCRT has very, um, uh, in a very, what do you say, in a characterized manner, they have taught you Mendelian inheritance and Mendelian genetics by dividing them into one gene inheritance and then two gene inheritance and things like that. I start with more of like a story like I did, okay? And then I go into the details telling you about which part of the story was the one gene inheritance and which part of the story was two gene inheritance, okay? Okay, so anyone has any doubt regarding this portion? See, one gene inheritance simply means when we are taking one gene at a time, which means two alleles at a time. And you also can call it as a monohybrid cross that we solve problems with. And it was very simple. You all understood the monohybrid cross and one gene inheritance, right? So give me a thumbs up. Okay. And no doubt then. Okay. Also, the concept of dominant and recessive is clear, because today what I'm going to teach you is associated with this, the concept of heterozygosity and homozygosity. So monohybrid, okay, Punnett square, everything is clear. So I'm also just going through the book and seeing what they ask you to read. Okay, anyone who has, so those who read, did you find any issue in this portion, which is currently ported on the screen? Yes, no, something? Wait, okay. In this portion, anyone had any doubts? So what does these lines say? So this is where you should read NCERT clearly. This is where questions are made from. So those who can speak, I think Aisha can, right? Aisha, can you speak? Can you read for me this paragraph from to this equation kind of a thing? Just read it and go slow so that we all understand what it is saying. Yeah.
From the Punnett square, it is easily seen that 1/4 of the random fertilizations lead to TT, half lead to TT, and 1/4 to TT.
It's
Through
all
the
it's all
F1
TT. So it's all TT. So you have to say capital T, capital T, capital T, small t. Or you can use the word homozygous, capital, heterozygous, and homozygous, small letters. Because if you go, this also has TT and this also has TT, it defeats the purpose of reading them. Okay. Do you understand what I'm saying?
Yes, sir.
Yeah. Okay. Cool. Yeah. So my video is spotlighted for all of you, just to let you know if any one of you wants to turn on the videos. Yes, Aisha, continue.
Though the F1 have a genotype of capital T small t, but the phenotypic character seen is tall. At F2, 3/4 of the plants are tall, where some of them are homozygous T while others are capital T and small t. Externally, it is not possible to distinguish between the plants with the genotypes capital T and capital T small t. Hence, within the genotypic pair uh, capital T small t, only one character T tall is expressed. Hence, the character T or tall is said to dominate over the other alleles, small t or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall, though the genotype is capital T small t, and in the F2, 3/4 of the plants are tall, though genotypically half are capital T small t and only 1/4 are homozygous T. This leads to
Homozygous
phenotypic.
capital T. It could be a homozygous small t also, right? Small t, small t. So
Yes.
1/4 are homozygous capital T. Yeah.
This leads to a phenotypic ratio of 3/4 tall, 1/4 homozygous dominant T uh,
Good. I
plus
like it.
half capital T small t and 1/4 homozygous recessive tall.
Homozygous recessive tall. How can it be recessive and tall? Yeah. Homozygous recessive tall.
That is
Yeah.
a 3:1 ratio, but a genotypic ratio of 1:2:1. The
Okay,
one
just stop. Just stop here. Just stop here. I just wanted to let you know that this is a classic example of how NCERT sometimes make simple things complex. Okay. So in very simple words, can anyone tell me what is this whole paragraph saying? It's just saying one thing. You could have made a Punnett square here and that would have made everything clear. So what it is saying, Iram, can you hear me? Or Parker, can you now unmute yourself? See, I'm discussing this because after this, I'm going to go into some complexities. So what it is saying in very simple terms, without using all of this. Someone is replying. Oh, I think there's some issue with Parker's Zoom software. It's okay. It's okay. Yeah, I saw that you left and joined again, but it's not solving the issue. Anyways, Iram, can you hear me? Can you speak? And not sure. Rab? Yes, Iram. Yes, we can hear you. Sorry, you are not able to hear me?
I cannot understand.
You could not understand the paragraph, you mean? Yeah?
Yes, sir.
Okay. Rabia? What about you, Rabia? Did you understand this paragraph at all?
No, sir.
No. Aisha, could you understand what they are trying to say?
Not really, sir.
Not yeah. So that is one reason why students, when they start reading genetics, they think, okay, genetics is not my cup of tea because I don't know what they're saying. So much of gibberish word. See, this whole paragraph is simply saying you one thing. They are saying they should have avoided writing, and this is exactly how they make questions also. Okay, in exams, they're simple questions, but arm twisted. You can hold the ear like this, but they will just go all the way around and hold the ear like this. So what they're saying is that 1/4 of the random fertilization, so fancy term, simply they mean that 1/4 of the progenies, when you cross two genotypes, which is one pure tall and one pure short, capital T and small t, when you cross it, just do it right now in your papers quickly in 20 seconds. Cross capital T with small t right now and tell me what happens in F1. Are you doing it, people? Just
Yes, sir.
yeah. Capital T crossed with small t. This is the parental generation. You will make a Punnett square because it is just a single genic inheritance, two alleles of the same gene. The Punnett square will only have 4. What happened? I can sense that someone got confused again. Just simply tell me what happened. Did you? So capital T, both the alleles are same. Yes or no?
Yes, sir.
Yes, sir.
And small t also, both the alleles are same. Yes or no?
Yes, sir.
Yes, sir.
So what I want you to do over this session and with me, I want you to be capable of doing most of biology in just your head. Okay. Though using pen and paper is highly appreciated, it reduces any chances of error, but becoming fast is also important in entrance. So whenever you see that both the alleles are same only, we know that these kind of parents or organisms can only form one type of gamete, right? For this gene, yes or no?
Yes, sir.
So if one parent can only form one type of gamete with respect to this gene, all the gametes will carry capital T only. And the other parent, which is small t, all the gametes will carry small t only. What will be the progenies? Do you have to go to a Punnett square for this? You can make a Punnett
No.
square, but when you will make a Punnett square, you will see that every box is capital T, capital T, capital T, capital T, capital T. Yes or no?
Yes, sir.
Yeah. Which means simply one gamete that came from one parent and one other gamete that came from another parent, capital T and small t, come together, and all the progenies, if in F1, are going to be tall. Make sense?
Yes, sir.
Simple. Now this paragraph takes from there and says that whenever there is a selfing between F1, which means two heterozygotes, there can only be one condition for heterozygote, a capital T and a small t. Am I right or not?
Yes,
This is
sir.
this is the only heterozygote condition in a single gene inheritance. Other two are homozygote conditions. Homozygous for dominant trait and homozygous for recessive trait. I hope you all can see my cursor, right? Yes, no?
Yes, sir.
Yeah. So the only heterozygous condition is this, capital T and small t. And in a heterozygous condition, if law of dominance is followed, which will always be followed unless they specify in the question. So if they write nothing in the question about any law, then all the three laws of Mendel are being followed. That will be our baseline assumption while solving biology. So if there is a heterozygous condition and there is no exception from any law, which of the allele will be expressed, capital T or small t?
Capital T.
Perfect. This is what happened in F1. All were heterozygotes. Now you are self-crossing the F1, a heterozygote with another heterozygote. Now do it quickly on paper. Cross a capital T small t with a capital T small t. Cross two heterozygotes together and make a Punnett square. Did you do that?
Yes, sir.
Okay. Does your Punnett square look something like this?
Yes, sir.
A tall heterozygote here, selfing with another tall heterozygote. So now, because both are heterozygotes, both can make two kinds of gametes. One gamete that carry capital T allele and the other gamete that carry small t allele. Make sense?
Yes.
Yes, sir.
And when you will make a Punnett square selfing, so this capital T will, so one progeny will be homozygous, dominant. Two progenies out of four will be same as parents, heterozygous. And one progeny will be homozygous but recessive, which none of the parent was. So do you see two combinations which are parental and two combinations which are non-parental? Do you understand that, everyone?
Yes, sir.
Now, in terms of phenotype, how will they look? How many of them will be tall?
Three.
Three. So these two will be heterozygous tall and this will be a homozygous tall. So three will be tall and one will be dwarf. And pay attention that this dwarf is coming from two tall parents. This is where we say that recessive traits skip a generation. Let's go one generation up. This is what you did in the first time, right? A
Yes,
tall
sir.
homozygous and a dwarf homozygous. You crossed, all were heterozygotes and all were tall. So this is parental generation and there was one dwarf. Then in the F1, was there any dwarf in F1 generation?
No.
No. Then in the F2, is there any dwarf again?
Yes.
Yes. So do you see the pattern that this dwarf, which is the recessive character, it was present in one generation, then skipped the other, and then again expressed itself in the third generation, like in the subsequent. So recessive traits skip generations. Does that make sense?
Yes,
Yes,
sir.
sir.
So this is one 4-marker question from boards. Just write it down. Justify and show diagrammatically. Justify and show diagrammatically that recessive traits skip generations. justify and show diagrammatically that recessive traits skip generation. If this question comes, all you have to do is to start here. You don't have to make the plant, okay? Just write the genotype, show the crossing, make gametes, and go all the way till here. So parent F1 and F2, and show that it was present in parent, the recessive character. It was not present in F1, and it was again present in F2. That's how genetics work. And just for doing this, you get the marks. But if you read the question in first go, you will think that, okay, I don't know how to justify that many times, right? So it's a difficult question with a very easy answer. And this is exactly what this paragraph is telling you. From a Punnett square, it is easily seen that 1/4 of the random fertilizations lead to capital T, capital T. It simply means that 1/4 of the progenies, when you cross two heterozygotes, will be a homozygous tall. Does that make sense?
Yes, sir.
Next, it says half lead to small t capital, sorry, capital T small t, which means one half, which means 2. 2 out of 4 is half, right? That's the confusing thing. Here they are writing 1/4. Then they're not writing 2/4. They're writing 1/2, which is exactly what it means. Like 2/4 will mean 1/2 only. So 1/4 is capital T, capital T. 1/2 is capital T, small t. And the rest 1/4, which is this one, is small t, small t. Okay. And then they're explaining that again in some, convoluted manner, that though the F1 have a genotype of capital T, small t, F1 means the first generation that we are selfing, though they are heterozygotes, but the phenotypic character seen is tall for them. At F2, 3/4 of the plants are tall, which you already told me. The phenotypic ratio is 3:1, right? You understand? It is just the play of words, where some of them are capital T, while others are capital T, small t. This sum of them is very ambiguous. You exactly know how many of them will be capital T, exactly 1/4, right? Of the total. But if the question comes, now I will dictate you a question. You will write that question and you will tell me the answer in chat. Write down the question. Everyone is ready to write the question?
Yes,
This is the
sir.
entrance. This is the entrance question, but I'll not give you options. So the question is, what fraction of the progenies, what fraction of the progenies sorry, what fraction of the tall progenies, I'm sorry, this is a tall word. What fraction of the tall progenies in F2 generation, will have a heterozygous allele makeup, will have a heterozygous allele makeup given that the parent genotype, given that the parent genotype is a heterozygous tall for both the parents. Given that the parent genotype is heterozygous tall for both the parent. And this question exactly came from this part, which you were finding very difficult when first Aisha read this part. But it's very simple a question. So everyone has written the question completely? Yes, no?
Yes, sir.
Now you have to give me the answer in terms of percentage. So the options are in terms of percentage. I'm not giving the option to make it will be easier then. You have to tell me the answer in terms of percentages, 10, 20, 50, 40, 90, 60, whatever. Do it. I got one answer in chat already. No, Aisha, read the question carefully. The devil and the divine both lies in the details. Aisha is very close but not correct. But good attempt. I appreciate you are fast, but sometimes being fast only help you if you are correct. Read the question again carefully. Read the question 3 times, 4 times. I want this. I want you to solve this on yourself. Come on, you all can do it. Come on. Write in the chat. I'm just telling you, it's very simple. All of you know the answer. Okay, I'll shut up for some time. Maybe I'm distracting you. We got one more answer in the chat. Iram, Aisha, and Rabia gave the same answer. Are like all 3 are wrong answers because all 3 of you have not read the question carefully. You all understood the concept. You all are solving it correct. But there's a twist in the question right in the first line. No, Mariam. Mariam also joins the clad. I'm giving the same answer. But I'm happy that you all understood concept. Okay, shall I discuss, or anyone is still trying for the glory? Anyone who's trying, just write that you are trying, and I'll give you one couple of more minutes. Otherwise, I'll go and do it. No, Mariam. Also no. Aisha is saying so 1 minute. Okay, I think Aisha is trying for the glory. Come on, Aisha, come on. Never give up. You know it. You got it. See, if I could understand this at one point of time, I think you people are way too smarter than me at any given, like of the age, the given age. I'm waiting for Aisha. Anyone else who's also trying? I think Mariam is now trying all the possible combinations. No, that's also not correct. Iram, that's not correct as well. Okay. Aisha? Oh, I have not received any. Aisha, what's your answer? You said 1 minute. It is 1 minute. Okay. Iram, would you like to read the first line of the question? Iram, are you there? no one speaks, and that's where it becomes very difficult for me. Okay, so there's disturbance at Iram side. Okay, Aisha, will you read? Disturbance is not a problem, by the way, because I understand we all are at our respective homes, so there will be some baseline, disturbance. It's fine, unless and until I
Okay,
can hear you.
so,
Yes, Iram. Yeah.
Okay. So what fraction of the top progeny in F2 generation
Stop right
will
there.
have
Stop right there. Read that line again.
what fraction of the top progeny in F2 generation?
Read that line again. Anyone got anything? Any tingle? Sixth sense? What they're asking you is, what fraction in terms of percentage or even in fraction? The question could also be what percentage? What percentage of the tall progenies in F2 generation? What percentage of the tall progenies in the F2 generation? Let's go back. In the F2 generation, you already know, if they would have asked what fraction of the progenies or the total progenies, then what you all answered in the first attempt was correct, 50%. 50% of the F2 progenies are heterozygous. But what fraction of the tall progenies in the F2 generation? They are excluding this dwarf progeny out. So you just have 3 tall, right? So this becomes your total. So what fraction of the 3, what percentage of 3 is heterozygous if you start with a heterozygous like parent to begin with? Aishas, you understand the question, everyone? Am I audible? Yes, no?
Yes, sir.
So if you are crossing these two and you are getting a generation, which means these two, which are the parents, are already F1, which are two heterozygotes, you self it, you get 3 tall, 1 dwarf. Now, what percentage of the tall progenies will be heterozygotes? So it will be 66%, approximately 66.6666. Is it clear, everyone? Does it make
Yes, sir.
sense? Yeah. So always read the question carefully. That's all there was in the question. And then this paragraph goes on to tell you what you already know. Okay, that within the genotypic pair, capital T, small t, only one character tall is expressed, hence it is called the dominant. And in the F2, 3/4 of the plants are tall, which we already know now. And though genotypically, half are capital T, small t, and only 1/4 are capital T, which again says that 3 are tall, but only half are capital T, small t, and 1/4 is capital T. This leads to a phenotypic ratio of 3/4 tall and 1/4 dwarf. Okay, so the tall will be capital T, small t, and the dwarf will be small t, as 3:1. But the genotypic ratio will be 1:2:1, because one will be the tall one with a different genotype, capital T. Two will be tall with a different genotype, capital T, small t, and one will be dwarf with a different genotype, small t. So is this paragraph still difficult for anyone?
No, sir.
Was there anything difficult in this paragraph at all, except for using too many t's and numbers? No, right? It was very, very simple. Does it make sense? Yes or no? Okay, now how many of you have math with biology in class 12? Anyone who has maths with biology? Iram. Okay, very good, Iram. Iram, would you like to read this small paragraph with this formula, please, for us? And also, if you wish, you can go on to give some explanation to explain us what's happening here. Don't worry about disturbances. We can hear you. Yep.
Yes, sir. The 1/4 is to 1/2 is to 1/4 is to ratio of tall TT is to heterozygous tall is to recessive dwarf is mathematically condensable to the form of binomial expression ax plus by to the whole square. That was the gametes bearing genes tall t or dwarf t in equal frequency of half. The expression is expanded as given below. Half tall t plus half dwarf t. The whole square is equals to half t plus half small t into 1/2
Yeah,
tall
same
t
half.
plus, yeah.
So basically what they are saying, okay, just don't read half t or that. How many, so Iram has math, but others, do you know about binomial expression? What does it means? Bi means two. Nomial is there are two terms, which it's expressed in forms of two terms, which are unknown, x and y. So ax plus by whole square. Okay. So this is your a, half is a, x is capital T, another half is b, and y is small t whole square. Ax plus by. So they are saying that the capital T and small t gametes are always present in equal frequency of half, which makes biological sense, right? In any parent, if there is a heterozygous condition, that is capital T and small t, what is the probability of a gamete getting a capital T? If you have two sides of a coin, you flip the coin, what is the probability of heads coming? Tell me. 50%, right? Half, 1/2. So, so gametes that bear the genes, capital T or small t, these are two different alleles of the same gene, they bear it in equal frequency of half. And if so, that makes A also half and B also half, because X and Y are your two different alleles. Okay, capital T and small t. And this binomial expression, ax plus by raised to power whole square, is written here. Half is a, t is x, plus half is b, and small t is y. So ax plus by whole square is equal to, it's just how you how you solve this binomial expression. Half t, it's whole square, right? So basically, what do we mean by whole square? Half capital T, you have to just multiply these two things 2 times, right?
Yes, sir.
4 square means 4 into 4. Simple mathematics. 2 square means 2 into 2. Now, if you take everything inside the bracket as one entity, let's say everything inside the bracket is z. So z square is equal to z into z. Is everyone following? Especially those who do not have mathematics. I want everyone's thumbs up if you understand, and do not do a thumbs up if you don't understand. Yes, Aisha, you have a question or you are raising hand? Iram understands. Aisha, do you understand or you have a question? You understand. Ravi understands. And Mariam understand. Khatun. What about Parker? Parker, you understood or you did not understand this? Okay, perfect. We'll go again over it. So pay attention, okay? And Iram, you will help me to help your fellow batchmate Parker understand this, okay? Okay. So what it says is, so you know about this ratio, capital T is to capital T small t is to small t small t. The ratio is 1:2:1, correct? This is the ratio, 1:2:1. Yes or no? So Parker, you have to keep writing certain things if you are, okay, you are still here, the same page. Now, how many total alleles are involved in this whole thing? There are three different kinds of expressions: a dominant homozygous, a recessive homozygous, and a heterozygous. But how many total alleles are involved? 1, 2, 3, 4. Just write that in the chat. How many total alleles do you see? 2. Perfect. Hiram also says 2. Very good. Thank you so much, Hiram, for your support. So there are two alleles. One is capital T, one is small t. Okay. And biologically, what is the chances of one allele in, so if it is homozygous, 100% chance of only that allele going in. So no problem, no clash in biology at all. But in a heterozygous condition, what are the chances of each allele going into a gamete is? 50%? Agree? Yes, no? Yes. Yes, Parker is on the same page. So that's exactly what they are trying to elucidate by saying that this whole ratio 1/4. So capital T was 1/4 or not? Let's go here. Capital T was 1 out of total 4. Thumbs up. Thumbs up will be a better idea. I think you can do thumbs up, right, Parker? Keep. Thumbs up is working or not? Okay, perfect. Oh, she clapped. Everything is working. Very good. So this is 1/4. What is this? Can I say it 1/2? Because it is 2/4, but I can just call it 1/2. 2/4 is 1/2. Again, a thumbs up. Capital T small t is 1/2 in this whole ratio. Yes. Capital T small t is 1/2. And then last small t is just like capital T 1/4. So this is 1/4. This is 1 out of 4, and this is 2 out of 4, which makes 1/2. Are you on the same page as with me, Parker? I'm waiting for a thumbs up. Yes, perfect. So simply, they are just telling you that this ratio 1/4 is to 1/2 is to 1/4. If they would have written 1/4 is to 2/4 is to 1/4, it would have been easier to understand. But since 2/4 is 1/2 only, it confuses some students. Okay. So they are just saying that this ratio 1/4 is to 1/2 is to 1/4, which corresponds to this genotype, can be mathematically condensed in the form of a binomial expression, which is ax plus by whole square. And how does this work? This works because x here is capital T and y here is small t. And a and b is the ratios, which is like, sorry, the frequency. And because in biology, both the gametes, both the allele have an equal frequency, half frequency to go into the gametes. The frequency is half. So half for capital T and half for small t. So we'll write it half capital T, which is ax, plus half small t, which is bx, and make it whole square. Everyone is on the same page till now? Parker, a thumbs up. Perfect. Now, what I am telling you is whenever we whole square anything, let's say 4 square, what does it mean? Does it mean 4 into 4? The same thing multiplied by itself is square, right? So whatever is there in this bracket if I just consider it as Z, or anything, Z, K, L, it will simply be, if it is Z, it will simply be Z into Z, right? Because it's square. Are you with me, Parker? I'm so happy to see all the yes. And I'm also happy that others are so patient when one person is learning in the group. Parker, are you on the same page with us? Do you understand why I'm saying this? I just need a thumbs up. Yes. So whatever is inside, the square of it will be the same thing multiplied by itself. So half capital T plus half small t multiplied by half capital T plus half small t. Make sense? Because it's square. Simple mathematics. And when you solve it, so how do we solve something that includes bracket and plus and multiplication? We apply the rule of BOD-MAS, BOD mass. You know that? Anyone? You must be knowing that rule in mathematics, right? Yep.
Yes, sir.
Yes, very good. So applying that rule, we will open the bracket and you will see first multiplication happens. So half T in half capital T, multiplication of the similar kind happens. BOD mass. BOD mass stands for bracket of division, multiplication, addition, and subtraction. So division, multiplication, addition, and subtraction. Since there is no division function as such here, so when we'll open the bracket, there'll be multiplication first and then addition. We can't add first. We can't add half T plus half T and make it. So it's like here, so it becomes 1/4 capital T, because these two come together, capital T. So 1/4 capital T, 1/2 capital T and small t. So this goes with this first, then this goes with other one. And 1/4 small t. It's simple. When you will just open this and solve, you will reach this solution. And this solution is exactly where we started with. Look here, 1/4 capital T, 1/4 capital T, 1/2 capital T small t, 1/2 capital T small t, and 1/4 small t, 1/4 small t. If you add all of this, what will, like, just add all the ratios, 1/4 plus 1/2 plus 1/4, what does it becomes? Quickly. 1/4 plus 1/4 plus 1/2, half. It becomes 1. Isn't it, everyone? Is it clear, Parker? Give a thumbs up. Yes, even Parker is writing the answer as 1. So the one who was not understanding gave the first answer as 1. I think it's clear in your head, right? So this is nothing to be afraid of. They are just simply telling you that Mendelian genetics was so mathematically accurate that even you could express and condense the Mendel's ratio using binomial expressions. And this is exactly why in the beginning, a lot of biologists could not understand. So they also could not understand. So it's not a big deal if you don't understand something. Ask it 2 times, 5 times, 10 times. There is never a bad student. There is always a bad teacher, is what I personally believe. Okay, so you should ask. And all of us are on the same page now, because from here we are going to take a leap shot and do something interesting. So this is how you have to read NCERT. It will take some time, but if you do it regularly in advance, like now itself, I completely agree that 1 night or 2 nights before the exams, you are not going to pay attention to this, because your mind will be in so much of a hurry that you will pay attention to the important concepts. That is where you will forget to, prepare these details, which actually make a lot of difference between rankings. So everyone knows 3 laws of Mendel. Everyone who will be sitting for, I'm saying that let's assume, if not everyone, 95% of the students will know Mendel's 3 laws of the Mendel's theoretically when they're sitting for an entrance. but 50 to 60% only will sit and understand this. And if a question comes on binomial expression, that which of the following Mendelian ratios can be expressed in the form of this binomial expression, and there are 4 options, you will just leave that option because you have not paid attention. And those who will attempt that and attempt it correct will get 4 or 2 extra marks, and that will just make a difference. Do you understand, everyone? Does it make sense? Yeah. Perfect.
Yes, sir.
Very good. So this binomial expression actually condenses, is a condensable form of the F2 generation genotypic ratio itself. Okay. So write down the binomial expression, which is this one, the binomial expression ax plus by whole square is a mathematically condensed form. The binomial expression ax plus by whole square is a mathematically condensed form of the F2 genotypic ratio of the F2 genotypic ratio in a normal monohybrid cross of Mendel. In a normal monohybrid cross of Mendel. And that's it. We prepared one question. Okay. Okay. Now, today I want to spend some time on 2 concepts, test cross and back cross. Okay. Everyone is clear for this part? Now you can just go and tell anyone this is super easy. Is it super easy or not?
Yes, sir.
But was it super easy in the beginning?
No.
You know why? Because you just looked at it, you looked at some expression, mathematics, oh my God. And that's where you lost the morale. You should never do that. You all, I'm again repeating myself, you all are smart enough to understand every single word written in NCERT. Okay. So just have that self faith and confidence and take anything heads on and see what can stop you from not understanding. This is simple English and logic. You can understand both English and logic. Done. Okay. Now let's come to test cross and back cross, 2 concepts which on which lots of questions are based in entrances. So when Mendel was doing these crosses, again, I'll just give you some quick 2-minute backstory of it. When Mendel was doing these crosses, first thing is there are many questions that arise from the cross. No one of you asked, how did Mendel know to begin with? We just very conveniently here, I'll go back to this thing. We very conveniently started the cross saying that let us cross capital T with small t, small t. But how did Mendel know that this is a homozygous tall and not a heterozygous tall with which he is starting the whole thing? If it was not homozygous tall, assume it was capital T small t. That will also look a tall only, right? All Mendel could see was just the height of the plant. Mendel did not know anything about genes at all. Remember? So no one of you asked this question that how do we know that this is capital T, capital T? All we see is tall. It could be capital T, small t also. Yes or no? Do you understand what I'm saying, people?
Yes, sir.
So how and why Mendel's cross always start with pure homozygous tall and pure homozygous dwarf? And how does he know that? Okay, let's do an exercise quickly. Let's see that you are Mendel. You took a tall plant and crossed it with dwarf. If it is a dwarf, it is for sure going to be a homozygous recessive. There is no doubt about it. So half of the problem is solved. But if it is tall, you can never tell just by looking if it is a homozygous tall or if it is a heterozygous tall. Now, let's say you were Mendel, you took a tall plant, but that tall plant happened to be capital T, small t, and you did this cross. So you crossed a capital T, small t with a small t, small t. Just do that cross right now in your notebooks quickly and tell me what happens. Tell me the genotypic and phenotypic ratio. Are you doing it, people? Yes, someone wrote something in the chat. Sir, can you repeat? Yes. So I am saying that let's assume Mendel took a tall plant and a dwarf plant. So the dwarf plant that he took for sure will be small t, small t. There is no other way you can make a dwarf plant here because law of dominance is being followed. But the tall plant that he took was not capital T, capital T. It was capital T, small t. Then also it will be tall only, right? We won't be able to make a difference between homozygous tall and heterozygous tall. Do you understand, Parker? Yeah, yes or no? Yes. So I am asking you to do this cross, keep taking a heterozygous tall with a dwarf. Capital T, small t crossed with small t, small t. Do it quickly. And allow me 1 second. Put this in charge. Yes, people.
Done, sir.
Done? Okay, so I received an answer. Okay, please give your answers. Just like you just write phenotypic ratio and genotypic ratio and just write the ratios. Yes, Parker, write the ratio. You wrote percentage, which is correct. I understand what you are writing, but write it in terms of ratios, like 3:2, 2:1:2, something like that. Iram, what about you? Did you understood what exercise we are doing here? Yeah, okay. I have not received your. Okay, and also let me tell you, when we talk about ratios, we take all the progenies into account. We do not. So if the ratio is 4:4, you cannot write it as 2:2, because 4:4 tells you that there were total 8 progenies, but 2:2 tells you there were total 4 progenies. So it's not the same as fractions. If it is 4/2, you can write 2/1, or you can simply write 2. But if it's the ratio, you have to write it complete. Yes, Parker. Very good. I'm glad that you are understanding this. So I have not received answer from, I've only received from Aisha and Parker. Rabia and Iram and Mariam. Just give a thumbs up if you have solved and you understand. That's what I want to know to move forward. Iram has understood. Okay, perfect. And what about Mariam and Rabia? Mariam is also understood. Rabia, waiting for you. Okay. Not listening. Okay, something came in the chat. You're not sure, but you gave an answer. I think, Rabia, you also made the same mistake that Parker made in the first. Did you um, just cancel out the ratios by something? Did you found that it's 2:2 and you made it 1:1? Because there can't be just 2 progenies, right? If you cross 2 parents, in a monohybrid cross, there will always be 4 progenies. In a dihybrid cross, there will be 16 progenies. So how can the ratio be 1:1? Yes, you also did the same mistake. Ratio tells you about the fraction of individuals in the total population. Do not cancel. If the ratio is 4:4, don't make it 2:2. Then it can be made 1:1. So which ultimately means from 8 progenies, you came to 2 progenies, which is a wrong representation. So the right answer is both genotypic and phenotypic ratio will be 2:2, isn't it? Yes or no?
Yes, sir.
Yes. So do you now figure out that there is a very easy way to test if a plant is homozygous tall or a heterozygous tall? You just cross it with a dwarf plant. If you cross a tall plant with a dwarf plant, and in the first generation you get all tall, it means it was a pure tall to begin with. But if you cross a tall plant with a dwarf plant and the tall one was heterozygous tall, then you will not get all tall. You will get half tall and half, which means 2 tall and 2 dwarf, right there in the F1 generation. Do you understand that? Yes or no, people? Give me a thumbs
Yes,
up if you
sir.
understand. Yes. So that's how Mendel also figured out where to start the cross from. He took 2 types of tall plants, crossed both of them with dwarf, and realized that in one condition, the F1 progeny is half tall, half dwarf. But in the other condition, all the F1 progenies are tall. So there can only be one logic behind it, and that was the logic that you just solved. That one tall was pure and the other tall was dwarf. So then he thought that, okay, let me start with pure parents so that I can understand the genetics properly. If you start with the dwarf parent, if you start with the heterozygous parent, you already have half of the allele as recessive, capital T, small t. So out of total 4 alleles, okay, look here, capital T, small t. Half of the alleles are recessive, half of the alleles are dominant. Make sense? Give a thumbs up. This is when you start as pure line. Okay, but when you start with a heterozygous tall, only one allele, which is this capital T, will be dominant. Rest 3 alleles, a small t here and 2 small t's here. So 3 out of 4 alleles will be recessive. And that is not a balanced way to start the cross. So that's how Mendel got to know how to select a pure line tall and not get confused with a heterozygous tall. So that's why we always start Mendel's cross with pure line tall and pure line dwarf, and then go on and see what happens. And only in this condition, the recessive character skips the generation. If you take a heterozygous tall with a dwarf, in the next generation only, you will see half of the recessive characters. All of this makes sense to you or not? Give a thumbs up.
Yes, sir.
Yes. So what we just did was what Mendel also did, and we call that as we call that as a test cross. So a test cross is a cross that you make to figure out the genetic composition of dominant characters, whether it is pure line dominant or heterozygous dominant. So what's written here is that, for example, let me read it for you. For example, whether a tall plant from F1 or F2 has capital T or capital T small t composition, it cannot be predicted. Therefore, to determine the genotype of all tall plant at F2, Mendel crossed the tall plant from F2. Remember, tall plant from F2. There are 2 different kind of tall plants in F2, right? One is pure line and 2 are heterozygous. So what Mendel did, Mendel crossed the tall plant from F2 with a dwarf plant. Okay. And this he called as a test cross. So This cross is done to test the genotype of a dominant character. So write down. A test cross is done between the dominant character of F2, a dominant character of F2 with a recessive homozygous condition. With a recessive homozygous condition. And why it is called a test cross? Right from next line. If the dominant character is homozygous, then all the progenies of the test cross will show the dominant character. Okay, capital T with small t, all will be capital T small t, which means all will be tall. So all the progenies of the test cross will show dominant character. But if the dominant character is heterozygous in F2, from F2, sorry, if the dominant character is heterozygous, which means capital T small t, and you are test crossing it with small t, then half the progenies will have the dominant character and other half will express recessive character. Like you just did and told me. Make sense, everyone?
Yes, sir.
Yes. So this is the representation that you are seeing here for a test cross. They are using not height here, but flower color as the criteria. So you have a purple flower too. Everyone pay attention here. You have a purple flower. I gave you a purple pea flower. And I am asking you, tell me whether this plant, which has this purple flower, whether this plant is capital W, capital W. Now here they have done the right thing. According to what NCERT follows for height, they use capital T for tall and small t for dwarf. But here they are using capital W for violet and small w for white. This is the right way to do it. We always use the letter which is the initial of the recessive character. Remember, I think I told you this, right? When we started discussing about this.
Yes, sir.
Yeah. So this is how it should be done for every. So even for, if you will read some good textbook, not textbooks, but some good books on genetics, you know, some, especially some international authors, when they talk about tall genes, tallness, the genes responsible for tall height of pea plant, they write homozygous tall as capital D, capital D. You can understand why, right? Because it's coming from the D of dwarf. Similarly, here they have written capital W for purple. Sorry, violet. So how will you tell me if it is capital W or capital W, small w? Homozygous or heterozygous? We don't know. So in both the cases, you cross it with a white flower plant. Okay. If you get half of them, sorry, here, half of them violet and half of them white, you know that the genotype was heterozygous. And if you get all of them purple, you know that the genotype was homozygous. Clear, everyone?
Yes, sir.
Perfect. So this is called a test cross. Now, one question is, what is a back cross? Write down this question. Write down the question, what is a back cross? What is a back cross? And the difference between test cross and back cross, I have seen multiple times coming in boards for 2 marks. Anyone who knows, I think in your school, this must have been done. What is the difference between a test cross and a back cross, people? Anyone who have done it, just. Test means to test a genotype. What does back means?
The background was not done.
Sorry, back cross is?
Not done in our school.
Back cross is not?
Not done in our school.
Not dominance. Okay, I'm trying to understand.
Back cross is not done in the school.
Oh, back cross is not done in the school at all. How? If test cross is in your syllabus, you just okay. They will ask you, okay. I don't know if it's then it shouldn't be asking boards then, but I have seen subjective questions like differentiate between back cross and test cross. That's weird. Anyways, so it's simple, very simple. Test cross is when you take a dominant character and you cross it with a recessive homozygous, because recessive will always be homozygous, to figure out the genotype of the dominant character. A back cross is when you take anything from F1 or F2, any progeny from F1 and F2 you take, and you cross it with any of the parent. So you are going back in generation. So to explain you, whether I cross capital T, small t with this, or I cross like any heterozygous here, capital T, small t, small t, small t. So when I go back in a generation, that's called back cross. So test cross is a type of back cross only. You understand or not? All test crosses are back crosses, but all back crosses are not test crosses. Everything that gold always glitters, but everything that glitters is not gold. Does it make sense to you or it's confusing?
Understood, sir.
Understood, right? So test cross is a back cross only because we are taking the heterozygous dominant from either F1 or F2 and crossing it back with the dwarf, which was actually the parent only, right? Parent was also homozygous dwarf to begin with. So you can just write down test cross is a kind of back cross. Back cross means going back and crossing anything from F1 or F2 with the parent generation. Okay, perfect. So far so good. Test cross, back cross, mathematical binomial expression of Mendelian genetics. Everything is clear? Yeah. Okay. So now law of dominance we have done, law of segregation. We also did incomplete dominance. This part I've already told you about, blood grouping and all. Then it starts inheritance of two genes where we came to dihybrid cross. We did one kind of dihybrid cross in the last class, right? Which was associated with law of independent assortment. We also know about this formula, correct? Okay. Just to tell you that even the dihybrid cross ratio can be mathematically condensed, but since that's a little more complex, that could not be just binomial expression. It has to be tetranomial expression. So it's not part of your course, but just know that all the work done by Mendel, all the ratios can mathematically also be verified. That is what is the genius of Mendel's work. Okay. Now, today I will just end with introducing you with chromosomal theory of inheritance. Okay. And then in the next class, we will draw chromosomes and we will see how exactly inside a cell chromosomes cross over and make recombinants. Okay. So now we understood everything that Mendel said. Everyone understood. Now everyone understands everything that Mendel said till now, all the three laws: test cross, back cross, mathematics behind it, law of independent assortment, linkage that we did in the last class, which is an exception for law of independent assortment, law of segregation, it has no linkage, it is universally true. Law of dominance, it has two exceptions, incomplete dominance and codominance. Everything is clear. Then only you will be able to understand this. Okay?
Yes, sir.
Perfect. Now, remember I told you that when Mendel published his work, it was around 1865. From 1865 till 1900, no one recognized it. Okay, everyone is like, I don't know what he's saying about. He's doing some mathematics, some weird ratios. This is not how biology is done. So we don't know what he's saying. But then also because Mendel did not know anything about, genes, gamete formation, etc., he could not give proper proofs. So his work was undermined. Then, after a very long time, so these are the reasons why we have already discussed this reason. But then, around 1900, the three scientists that we talked about, Hugo de Vries, Carl Correns, and von Schermack, independently rediscovered. And by that time, microscopes were a common thing. People were using cells, people were observing cells under microscope, and they already had some idea now about chromosomes. Now, again, what are chromosomes quickly? Anyone? Iram, Aisha will be. Speak. What are chromosomes? Quickly, come on, you know this. What are chromosomes, people? Chromosomes simply are condensed form of the DNA inside a nucleus. Okay. Yes, Parker says thread-like things containing genes in the nucleus. Yes, that thread-like thing, you're right. Chromosomes also look like thread, but they are condensed form of even thinner threads called DNA. So DNA comes together and form one chromosome. Okay, along with protein, it is super coiled and condensed. So people could see chromosomes in the cell. That's called karyotyping. You can actually stain a chromosome. So people also develop, scientists also develop by that time, stains to put on the cell, and that will mark the nucleus, and inside it, it will mark small bodies. The word chromosome itself came from there. It has no roots in DNA or genetics. The word Chrome, you use Google Chrome. Chrome is called Chrome because the whole logic and the logo of Chrome is about colors. It has 4 colors in its logo, 5 maybe. So the word chromo or chromatography, chromatogenic, or your photochromic lenses, they're all called, chrome means color and soma means body. So in biology, chromosomes technically means colored bodies. And they were called colored bodies because people could put some stain on the cells and color these chromosomes like black or whatever, indigo blue, whatever the stain color is, and you can see it under a microscope floating. So people also started understanding chromosome movement during meiosis. So there were two people, very brilliant scientists, Walter Sutton and Theodore Boveri. They were working on cells, and especially they were working on nucleus of the cells. And they were looking at these chromosome behavior, how chromosome goes from one cell to another when the cell divides in meiosis. And they realized that the behavior of chromosomes is exactly the same of what Mendel told about the behavior of genes. So they saw that just like genes segregate independently of each other, and the alleles of genes segregate independently of each other and go into different gametes, during meiosis, when gametes are formed, chromosomes also move like that. So look here, G1, G2, and then the phases of meiosis starts. So they are only taking anaphases to just explain you that, do you see at G1, this yellow chromosome was with red, orange with green. So there are basically 4 chromosomes, let's say. a yellow pair, a red pair, a orange pair, a green pair. How much time we have? Okay, we are out of time, but I will just tell you this. So then from G1 to G2, replication happens, DNA duplication. And you see that now there is a centromere and there are 2 copies of each chromosome. Okay, they have just made it small and big to give you a sense of different chromosomes. So one is yellow, one is red, one is orange, one is green. It duplicated. Now there is a centromere, but still there is one chromosome of yellow having 2 chromatids, one chromosome of red having 2 chromatids, one chromosome of orange having 2 chromatids, and so on. Then at meiosis 1, yellow and red got separated from orange and green. So one kind of cell only gets all the yellow and all the red. Another gets all the orange and all the green. Now let's see. Yellow is the gene for height. Red is the gene for eye color. You know, we are drawing parallel between genes and chromosomes. So according to Mendel, this will also happen for genes, right? The gene for eye color can independently segregate from the gene of hair color, given that there is no linkage. So in normal condition, we are not talking about any deviations or exceptions. But it does not stop here. Mendel also said that even the alleles of the same gene can segregate independent of each other. And that's exactly what happens at meiosis 2 anaphase. These 2 yellow chromatids from the same yellow chromosome and these 2 red chromatids from the same red chromosome also get separated from each other. So not only that chromosomes get separated independent from each other, the chromatids also get separated independent from each other. It is exactly parallel of what Mendel said. Genes get segregated independent of each other and the alleles of one gene get segregated independent of each other. Does it make sense, everyone? Yes or no? Give me a thumbs up. Are you already bored and sleeping? No, right? Parker, I'm trying my level best, but no, it's difficult for me to look at a screen and teach. But I hope you all are understanding, right? Everyone is understanding? Aisha, Rabia,
Yes.
yeah. So do you see some commonality between how chromosomes are inherited and how genes are inherited? There is a parallel similarity, right? So that is what Theodore and Boveri figured out. And they gave a theory, which is known as theory of chromosomal theory of inheritance. They did not talk about factors or genes. They said that, okay, chromosomes are the things that get inherited. And they were right, because chromosome is, now we know after 100 years that chromosomes are the things on which is made up of DNA, and on DNA is present the gene, and 2 different versions of gene are called alleles, and basically this is how whole genetic works. So these earlier scientists were brilliant enough to understand that what Mendel did with saying genes and factors, chromosomal theory of inheritance also makes means the same thing. So it was given by Walter Sutton and Theodore Boveri. I want you all to just read and this part, table 5.3 downwards. So it is page number, I don't know, page number is this. Sorry. What page is this? There's no page number here.